My project is titled An Investigation of Bacteria Filtration with Activated Charcoal. And essentially, it looks at the relationship between two different materials, bacteria and activated charcoal, and specifically how they interact when they are in the presence of one another. Now, if we take a trip back 7 months, the entirety of this project was summed up with a news article. In this article, it documented our ability as a society to treat different diseases, infections, and viruses, and specifically looked at how this treatment has changed over the course of history. Now, this article caught my attention, and I decided to do more research. Now, through this research, I found that the key aspect of treating bacterial infections is the idea of an antibiotic. Now, of course, the purpose of an antibiotic is to get rid of an infection, but it can do so in two major ways. The first is killing the entire population of bacteria, but the second and more common is to weaken that same population to a point where the host's natural immune system can fight off the remaining population. Now, in order to do either of these things, it looks towards two main pathways to kill bacteria cells. Now, the first is the destruction of the cell wall. Without a cell wall, the cell dies. The second is preventing replication of the bacteria population, in a sense making it smaller and smaller until, again, the natural immune system can fight it off. Now, the use of antibiotics has increased tremendously over the past few decades. But in the last decade, the idea of antibiotic resistance has unfortunately also surged in popularity. Now, antibiotic resistance occurs when a particular strain of bacteria is no longer subject to the effects of a certain antibiotic treatment. Now, doctors and physicians all over the world will disagree on exactly how resistance has occurred, but one of the main ones is individual error, when somebody doesn't take the proper dosage of the antibiotic treatment for their infection. Now, with antibiotic resistance, the reason it is occurring is because antibiotics look at the chemical nature of bacteria. They try and do something to manipulate the chemical structures within the bacteria cells. Now, clearly this is failing. So I hypothesized that we instead need to look at something that relies on physical characteristics, which is how I came across activated charcoal. Now, I knew that activated charcoal was something that was in my fish tank filters for years, and I knew that these black particles somehow cleaned the water. But I didn't really know how exactly, so I started doing some more research, and I found that activated charcoal has very small micropores that are able to trap contaminants. With all of this information, I created my testing purpose, which was to provide a long-term treatment method for bacterial infections with activated carbon. Now, with all of this research and my testing purpose, I started to design the actual testing itself. Now, I designed it into two phases. The first one looked at activated charcoal and bacteria growing in the same environments, and the second looked at activated charcoal's ability to filter out bacteria from a source. Now, for phase one, I had to start with reactivating a sample of freeze-dried Micrococcus luteus, which is a biosafety level 1 bacteria that is common for skin infections. And then I grew it in two different environments. The control, which was a petri dish of plain agar, and a treatment group, which was agar with 5 grams of activated charcoal. I had 11 petri dishes in each set, and then I allowed them to grow for 24 hours at a temperature of 25 degrees Celsius. After that 24-hour time period, I compared the relative growth sizes, and I gave it the classification of no growth, small growth, medium growth, or large growth. This table summarizes all of the results, showing that each of the control groups showed some amount of growth, while none of the experimental group showed any visible growth. With all of this data and results, I moved into phase 2, which again looked at the actual filtration of bacteria. So again, I reactivated another sample of M. luteus, but instead of growing it on petri dishes this time, I grew it in a nutrient broth. So I allowed cultures to grow in a broth for a period of 48 hours until the broth became cloudy, which is how you know that there was bacteria in that sample. I then filtered it through 10 grams of activated charcoal, which were housed in a normal grade coffee filter, and then I measured the absorbency units in a spectrophotometer. So all of this table shows the different absorbency units that were given after filtration, and I compared those to several different controls that were the sample before filtration. This is the differences summarized, and I found the average, the standard deviation, and I ran all of my data through a two-tailed t-test with unequal variance, and I obtained a p-value of 1.289 times 10 to the negative 8. So when you have a p-value that small, you start to get really excited, and I used this statistical test to summarize my conclusions and look at what all of these results were overall proving. So from phase 1, all of the data was qualitative. It was all based on visual representation of growth rates. So unfortunately, no definitive conclusions could be drawn from that, but it did show that activated charcoal can influence the growth rates of bacteria in some way. As far as phase 2 goes, this provided the quantitative data that phase 1 was missing, in which I could find statistical significance that activated charcoal was able to remove bacteria from a contaminated source and lower the amount of bacteria in that source after filtration. All in all, what this means is there are several real-world applications to using activated charcoal for bacterial infections in both preventative care and curative care. So I'm really happy with the results that I was able to gain through this experimentation, but with any experiment, I would love to conduct further testing to try and get more data and strengthen these results. Specifically, I'd like to look at changing different variables, such as bacteria types, the amount of activated charcoal used, and the amount of time that I allow the bacteria to sit and be filtered through the activated charcoal. And I'd also like to look at the medical effects of using activated charcoal, so how it acts with skin contact, what it looks like when you use it internally versus externally. So I would like to continue testing down those routes, but all in all, I am very happy to say that I believe activated charcoal can be used to treat bacterial infections that will provide a long-term treatment not subject to the resistance.